Thank you very much, Pavlos. I'm very glad I could be here today. And I don't want to compare California and Boston, but if you come visit us, you will not have multiple flights canceled due to ice on a runway. I'm just saying. So I'm going to give a very different sort of talk than some of what I think you've seen today. The point of this talk is going to give you a big picture overview of the problems that people are applying machine learning to in chemistry and the types of models they're using, what the questions are. And the goal here is that you'll find these so interesting and you'll have the right keywords to go and start doing some searching, learn more about these, and to work on them. But why am I an interesting person to do this? So as Paulus mentioned, I'm part of this Google Accelerated Science team, and our goal is to bring together computational folks with domain scientists to do something interesting, to find some interesting science. And my training is all from the AI, machine learning, data science side. And it's only in the last 5 years that I've really gotten back into the physical sciences and looked at everything from chemistry to agriculture. And chemistry has been a particular interest of mine, so some of the work I'm going to talk about today is work that I've done in collaboration with others in my group, but I'm also going to refer to a whole bunch of other work that's gone on across the field. So don't feel like you have to write down all the references for all the things. At the end of the talk, I'm going to have a bibliography with all of the links to all of the stuff that I talked about today. So the first problem I want to talk about is molecular property prediction. And the basic idea here, and people have wanted to do this for a really long time, I have some molecule and I want to predict some property of it. So if you don't know this molecule up here, if you don't recognize it, this is actually aspirin. Hopefully you've used this when you've needed it. And people have for a long time wanted to do things like, is this molecule going to be useful for something? Is this going to be soluble in water? Is it going to treat some disease or some condition? Or is it going to be toxic to the body in some way? These are all, these kinds of questions people have been asking for a long time. People have been talking about cheminformatics for a while. And you also hear these things like virtual screening and property prediction. And typically you want to do this because these types of things can be very expensive to compute in a lab, right? So if you have to make the molecule, do some experiment, if you can build some kind of machine learning model, then this can be a much better way, much faster to go and explore chemical space, to explore possible molecules. But there are cases, and one of the recent things that happened over the last few years are a bunch of uses where we're not talking about things that are done into a lab. You've heard a little bit about kind of physical calculations like quantum chemistry calculations. And one of the interesting statements of the world is even though we understand so much about what's going on at an atomic level, the theories are very, very difficult to use. And so a typical kind of quantum chemistry screening program would run calculations that are kind of minutes, hours, or days per molecule. And very quickly, you can exhaust even the resources of very well-funded institutions. And so recently, ML has become a big part of how people are trying to do these kind of quantum chemistry screenings. So the idea is, instead of running the quantum chemistry calculations on all of the molecules you might be interested in, you only run it on a part of them. You make some training set, you run those hour or day-long computations per molecule for a small set of molecules. You then get the values for those molecules for that training set. You build some ML model, and I'll talk a little bit about how people have approached this. And then you can use that ML model, and typically this will run in milliseconds or seconds, and you can scan over a lot more of chemical space. So you see both people applying these kinds of models to these sort of real-world experimental data, as well as to approximate these expensive calculations. But there are a couple of interesting considerations you should think about how you're actually going to build ML models for this. One is, I sort of drew a picture kind of like this up here for molecules, this 2D picture. And this is pretty common. If you remember from your chemistry classes, you've probably seen these kind of things that represents how the atoms are bonded with each other. But in reality, these things are 3D structures. And often, there can be multiple 3D structures. The molecules can often be somewhat flexible or adopt slightly different conformations. And so in some ways, that 3D picture is more true to the reality that's actually going on. But sometimes you don't know what it is. And 3D actually gives you another interesting challenge. So as you can see this molecule moving around up here, the molecule is exactly the same no matter which way you were looking at it from. And so actually coming up with models that are appropriately invariant to your frame of reference, to what way you're looking at the molecule, has actually been somewhat of a challenge. And you will still see people publishing new ML models that actually don't have this property, that are not invariant to your frame of reference of the molecule. Another interesting problem that comes up is, if we consider a simple water molecule like this, typically when we're encoding these things for a machine, we have to make some arbitrary choice about order. Like in the picture on the left, I said that hydrogen is atom 0. But I could have just as easily called that oxygen atom 0. And ideally, you want a model which doesn't care which one you have. And again, when I say it like this, you're probably sitting there saying, well, of course I want that. Why would I ever build a model that doesn't do that? But we actually spent a while building models that did not have this property. And a number of representation of molecules have been suggested which didn't have this property. And it's very easy to accidentally get this wrong. So there is another funny thing that comes up with 3D that is another confounding problem. So there's these things called enantiomers. So here I have two molecules. These are two different forms of ibuprofen. And if you're very good with your 3D sense, you will recognize that these are kind of like your right hand and your left hand. There's no way to move your right hand around and make it exactly line up with your left hand. They're essentially mirror images. But a lot of representations of molecules, if you're only looking at pairwise things, you actually can't tell the difference between these. They look exactly the same when you look at things like pairwise distances, but they can act really differently. The S form of ibuprofen here is the only one that is actually therapeutic. The R form doesn't do anything until it is converted into the S form in some way. So sometimes this matters for some applications, is really important, and sometimes it's not. But as soon as you start thinking about 3D problems, you start running into interesting complications like this. So what I want to do now is kind of give you a really quick run through of the strategies that people have used for building machine learning models on molecules for this kind of property prediction. And the most kind of traditional way to do this is to come up with some featurization algorithm. The idea is you take your molecule, you're going to run an algorithm and convert that molecule into a simple representation. The example I'm going to give here is from something called extended connectivity fingerprints. This is kind of I would say kind of the workhorse of cheminformatics nowadays. This is the most common featurization that people use. And the idea is pretty simple. You look at individually at each atom in the molecule. And you first of all, you consider just that atom, and you say, okay, I'm going to take that atom, that's one fragment. And then I'm going to go out one step, and I look at all the atoms next to it, that's another fragment. And then I'm going to go out another step, and that's another fragment. And if I put all those fragments together, I put them through some hash, I can very easily end up with this fixed length binary representation, which represents all of these fragments in the molecule I've created in this way. And the nice thing, the reason people wanted to make these fixed length binary representations is a lot of the traditional ML algorithms that people created only really worked on fixed length representations. People make comments saying, oh, all ML algorithms require fixed length representations. It's not actually true anymore, but it was actually true for quite a while. So the things you've probably heard of, like random forest and SVMs and logistic regression, all assume you have some fixed length representation. And so this has been a very traditional way to do things. One of the things you can do with those simple featurizations, and also with a little bit more complicated featurizations, is to just define a similarity function. So either using that featurization or some other method, just say, I'm going to define a function, and we usually use K because these are kernels, saying how similar molecule X and Y is. And then the most common thing that people do today, especially in these approximations of quantum chemistry calculation, is to then use something called kernel ridge regression. And the idea here is actually fairly simple. If I want to predict the property of some molecule, here I have aspirin back up here, all I do is say, well, how similar is it to every molecule that I know about, every molecule in my training set? And the part of the ML is just learning these weights alpha that I have on there. And there's a well-defined way to actually do this. And so, here, the nice thing is the ML part is fairly well understood, and a lot of the complexity goes into how you actually define that similarity function. And people are still going through and creating more and more similarity functions for use in molecules in all kinds of different ways. So let's add some neural nets to this. I'm from Google, you're probably not surprised. I'm going to talk a little bit about neural networks. The first approach that really used neural nets in a serious way for these problems is exhibited well by these things that are often called Baylor-Parinello networks. So here the idea is I start off with my atomic positions, and then I'm going to compute these things called symmetry functions. And the idea with the symmetry functions is all those invariances I want to, what orientation I'm looking at the molecule or the ordering of the atoms. You have the symmetry functions such that they are symmetric to all those transformations. So it doesn't matter what order I present the atoms in, you get the same symmetry function out. And that's how you get all of your invariants. And then once you have that representation of each atom as this set of computed symmetry functions, you go through your traditional fully connected feed-forward neural network layers like you've seen mentioned several times, and then out comes a prediction of something like the energy of that configuration. These things are very, very commonly used when you're looking at 3D configurations of atoms because the symmetry functions are typically defined on 3D positions. But really, this is a pretty simple application of neural networks. There's actually not that much structure to these neural networks. All we're doing is running these fully connected layers on each of the atoms. Much more interesting things have been done with more complicated models. One of them is represented by this picture from a group that developed something called AtomNet. So we've heard several times about convolutional neural networks to recognize cats and dogs on YouTube. We know a lot about those things. You can extend those same ideas to 3D. And so what this is showing here is that little box is showing part of this convolutional network that is recognizing a particular chemical group. And this is something that the model learned how to do. It learned that it was relevant for this particular protein. The kind of ribbony thing up there is representing a protein that this small molecule is going to bind to. Now, this is sort of a very natural extension to the 2D convolutional networks. The problem is that the scaling starts to really kill you. As you go to 3D, especially at the resolution that we really need for atomic systems, the scaling here can be really poor. And so figuring out either how to scale these appropriately or to build them in a way that they don't scale in the same ways is kind of the interesting challenge with this approach. The other kind of main thread of neural network approach on molecules are these things called graph networks, and you also hear this term message passing neural networks. And I put some math up here just to make sure you guys would take me seriously. But really all that's going on here is the idea is that at any point in time, you say every atom, every node in this graph knows something about itself. And from what it knows about itself, for example, it might start off knowing, oh, I'm a carbon atom. That might be the only thing it knows at the beginning. It sends a message to all of its neighbors. And that's this message function up at the top. Those neighbors get the message. They then update what they know about themselves. And you keep doing this again and again and again. And you iterate this many times. And as all these nodes pass messages to their neighbor, they sort of learn more and more about their chemical environment. And as they learn more about their chemical environment, then you can eventually use that to make it a useful prediction. And people have made many, many variants of these kinds of networks. I will say they are some of the more popular ways to do neural networks for small molecules. And you can put in either that 2D just bonding structure, or you can put some 3D information in there. The other really interesting thing that's happened recently is really taking advantage of 3D geometry. So some of you may recognize these things I have up on the screen. These are the spherical harmonics. If you remember your quantum mechanics, you'll remember these from the hydrogen atom. But these are also really useful mathematical constructs because they're an orthonormal basis for functions on the sphere. And so they're really useful mathematical objects. And so what 3 different groups sort of co-discovered over the last year or so is that you can encode in a neural network using these spherical harmonics in a way that you guarantee you get certain properties out. Like if I rotate the molecule, all of my predictions rotate in exactly the same way. You can get this very nice geometric understanding using these kinds of functions. I decided not to try to encode a lot of the math that actually has to go into this. This is actually a very interesting approach, and if you read these 3 papers, you will see 3 fairly different ways to formalize this same approach. But I think it's a very interesting direction that's going on, is really taking advantage of the fact that we can encode very complex operations in these deep neural networks. So let me mention a few kind of extras in here, a few variations on this basic idea of predicting some number on a molecule. One of them is that sometimes people use this to predict something about each individual atom rather than about the whole molecule, like predicting forces or predicting something about the charge of the individual atoms, et cetera. The other kind of things that's going on is rather than just predicting one number, you can predict a much more complicated output. And here I've sort of represented the idea of, given a molecule, can I predict its mass spectrum? What would happen when I put it into a mass spectrometer with a particular set of things? And you see things like this going on as well. The other thing I want to mention here is energy is kind of special. Right? If you talk about the energy of the geometric arrangement of the atoms, this is something that a lot of people care about. It relates to if we want to do things like molecular dynamics, if we want to do things like reaction prediction, et cetera. And the little illustration over on your left here is showing the types of transformations that we need to understand what is the energy of that particular configuration. And so a lot of work has gone on to this, and you'll hear terms like machine learned approximation potential or machine learned force field. And the idea here is you can take a much more accurate quantum chemical calculation, build a machine learning model on that, and then run that much, much faster than you could do those very, very expensive calculations, so that you could do things like run MD, and et cetera. And so a lot of work has gone especially on this problem. Now, there's a couple things I want to highlight here, and these are actually good general questions, but they've come up especially in the context of molecules. One is thinking carefully about what you actually want your machine learning model to generalize over. Because if you want your model to generalize only from the configurations of one molecule to other configurations of that same molecule, you should build a different model than if you want it to generalize from one molecule to another one, or from smaller molecules or bigger molecules. And how you ask that question, it very much affects how you train your ML model, how you think about the problem, the questions you should be asking. The other important thing to think about is what information do I have to start with in my problem? And this is actually, I think, well encapsulated by something you see in a data set called QM9. It's one of these standard benchmark data sets for testing molecular machine learning. But what's interesting about it, they had basically what this data set has is, for a given molecule with its geometric arrangement of all the atoms that's given to you, what is various properties like energy and heat capacity and polarizability, et cetera, these things that you can compute with quantum chemical calculations. But what's interesting is the geometry that you get, that is given to you in that data set, actually comes from a fairly expensive quantum chemical calculation. And so you can't actually take the model that you learn from this QM9 data set and do anything with it on anything you don't know about, because you have to get that same geometry. And so that geometry problem, if you solve that problem, you've already done most of the work. And so this is something where if you don't think really carefully about what data you have and what data you're going to have when you want to actually use the model, you can confuse yourself very quickly. The other thing to think about is very often, especially machine learning types, will sort of settle and go back to the standard error functions they know about. You'll hear, oh, root mean square error. That's what I always use. Let's use it here. But very often, what you actually care about might not be that. If you're predicting energy, for example, you may not care about the absolute energy, but you may only care about the difference in energy between two configurations. If you don't think carefully about these problems, you're likely to confuse yourself, not just in molecular machine learning, but in a lot of machine learning problems. So let me go on to a somewhat different topic, and this is somewhat more modern compared to the stuff that I've been talking about. So before I said, okay, the goal of the property prediction was, given a molecule, let's predict some property of it. Generative models just flip this around. They say, given some property I want, what molecule should I have? Which molecule should I create? And these are typically called generative because the output is this much more complex structure than just a single number or a small set of numbers. Now, what typically happens is not that you actually have a function here, but these generative models are typically probabilistic. What they're saying is, that given some property I desire, I have some probability distribution I'm sampling. Over molecules, and I can keep sampling to get more and more molecules. So why is this interesting in chemistry? So what I've shown here are two different, very simple illustrations of sets of molecules. The one on the left here in the dark colors, these are molecules randomly sampled, knowing only the most basic thing about how atoms bond. Carbon should have 4 bonds, oxygen should have 2. And says all I'm going to do is obey those very simple rules, and I'm going to sample molecules. The ones on the other side, these are molecules that that chemists have actually done something with. These are in this repository called Kimball. Now, if you have any chemical knowledge at all, you will look at the ones over here and say, many of those just look crazy, right? There's no way those would actually exist. But actually defining what is it about those molecules that makes them look crazy to a chemist, it's not actually obvious. And this is where generative models are kind of interesting. When they're capturing this this essentially probability function that's going on inside people's heads saying, that's the type of molecule I want. That's a reasonable molecule, but I can't write down the rules to express it. And so this is one of the kind of formulations you typically see in generative models, which is, hey, all I'm going to do is I'm going to capture some complicated distribution. That's this top example I've said here, saying, given nothing, just generate me a molecule, kind of like what I've seen in Kimball, for example. Generate me a useful one. And this is one of the types of things that people do, just to say, let me see if I can just generate a bunch of reasonable-looking molecules and see what we can do with them. The other variant you typically see is, okay, I've already got some reasonable molecule. This is the core of a steroid. Many of you probably have run into those in some way. And I want to figure out some elaboration of it, some modification to this core, and because maybe I want to use it for further exploration or for deciding new molecules to make, et cetera. And so that's another common formulation for these generative models to kind of support this kind of search effort. The other variant you can get here is, instead of starting from nothing, maybe you know something like, oh, I want it to bind to this protein. I want it to bind to this particular target site. And people are starting to build models that generate directly from something about the information of what they want this molecule to do. The other variant here is, I was saying, oh, let's predict 2D pictures. That's what I was showing up there. You can also obviously predict 3D. The other interesting variant here is starting from a 2D configuration of the molecule and generating the 3D geometry. Now, for the particular case I've shown up here, it's actually not that interesting. This particular molecule really isn't that flexible. But you start to get to much larger molecules like proteins and so on. This can be a really useful process if you're trying to predict things like protein structure, like Lucy was talking about, or even things like chemical reactions and how molecules will change over time in the presence of other molecules. A couple other things about how people think about this. There's kind of a big split in how people build generative models. One of them is to use these things called smile strings. Smile strings are these textual representations of molecules, and they've been around for a long time. They're a very, very useful way and a common way to talk about what molecules are. And the nice part about this is we've done a lot with building language models. This is what a lot of the folks from the CS side have done, have built language models. And you can kind of obviously go and say, hey, Smiles looks like a language. Let's run our standard language models on it. The other big approach in these generative models has been to use some of these graph-based models, like these message-passing neural networks I was talking about. Now, I have to say, I actually have a strong bias towards the graphs being the correct way to do this. And one of the reasons why is actually illustrated on this slide. If you were very, very quick and you know the smiles grammar really well, you've probably realized all of those smile strings actually all represent vanillin, right? This is one of the things that gives vanilla its distinct flavor and smell. And so all of those are equally valid ways of representing exactly the same molecule. And so when we ask our models to learn smile strings, they also have to learn something about all these different ways of expressing exactly the same thing, which seems like, to me, kind of a silly thing to ask our models to learn, in addition to the chemistry we want them to try to actually understand. Now, that being said, there actually have been some very useful models built with smile strings, but it still surprises me a little bit that they work as well as they do. So let's talk a little bit more about what you might want to do with this, or how these models really work. There's kind of two main strategies, again, that you see even on top of what I just described for how you would build a generative model. One is to have one of these encoder-decoder networks. The idea is, given a molecule, you map it to some point in high dimensional space. For the purposes of my slide, high is 2. So I have a 2-dimensional space of molecules on here. And then every point in space you can then decode into a particular molecule. And the idea is that, then in some way nearby molecules that are nearby in this space are somehow related. And this is, we often talk about this as learning a latent space for these molecules. The other main strategy is just to define transformations between molecules. What I've shown on the other side are the transformations that we defined for some recent work we did for cyclohexane. So if you start with that black ring, what are all the sort of nearby molecules you might make from this? And you can, in that way you can define how molecules are connected to each other. And other people have defined very similar things. So why would you do it this way? What do you want to do with these things? So in both of these, you're often thinking about this in terms of doing some kind of property optimization, doing some kind of search. So the hope in this latent space world is that when I've mapped all these molecules down into this latent space, I can then walk along that latent space and use some of my standard optimization techniques, doing gradient ascent, doing random restarts, pick your favorite optimization algorithm, and I can run it in that latent space rather than having to think about molecules as I was before. The other strategy is using those kind of those graph transformations I talked about. This very quickly fits into one of these reinforcement learning paradigms. Now, this reinforcement learning may be a thing that you've heard about recently. It's been a technique in AI for a really long time, but it's one of the things that's gotten a bit of a renaissance recently as we've developed a bunch of techniques that help this work better. But the basic idea is reinforcement learning helps you learn what actions to take. So here you're learning what actions do I take, what modifications do I make to this molecule in order to make it better in some way. And this is showing a particular optimization path for this toy problem of optimizing a property called QED. So there's an interesting problem where both of these things kind of come together, and this is coarse graining. So what I'm showing here is a small part of a protein where MD has been run to show this thing folding and unfolding and kind of gradually finding a stable position. And and hopefully Lucy has convinced you this is an interesting problem to try to figure out how these things fold. And people have been worried about this for a long time. One of the big approaches in these physics-based approaches is rather than having to model every single atom on there, as you see I've got, there's lots of little dots up there representing all the different atoms. Maybe I could do this in a simpler way. And people have proposed a variety of different simplifications of the protein. Here in these pictures, it's drawing these various orange balls, or beads they're sometimes called, to simplify that computation. And that way you don't have to run all the computation on all the atoms all the time. But and in fact, there was the Nobel Prize in Chemistry a couple of years ago was awarded in part for people developing these kinds of coarse-grained models. Turned out there are actually 3 different problems that come up when you start thinking about coarse graining like this. One of them is, how do you actually do the coarse graining? As you saw in this previous slide, there are actually a lot of different options that people have come up with, saying of different ways of creating a coarse-grained model for the same protein. And how exactly you should do that is not obvious, especially when you get to things that are a little less regular than proteins. If I'm start talking about other kinds of molecular systems, where's the appropriate place to put the grain? What kind, how should that actually work? That is one of the interesting problems that people have been working on. The other one is, given the coarse graining, I need to have some force field. I need to have some energy, a distribution of energy for a given configuration. What I've shown here is, ideally, in some cases, you would like for it to be a smoother thing, easier to optimize, easier to search, easier to do MD on, while still being faithful to the original all-atom calculation. And then the last problem you have is that there's this problem of fine grain, which is given the coarse grain configuration, you need to then back out and say, ooh, what is the actual all atom configuration? Sometimes you need this to do further calculations or to match with experimental data. So I'm sure you won't be surprised when I tell you where I think ML is going to be useful to this problem, which is everywhere. And right now, we're already starting to see people apply ML to all three of these different problems. And so as you can see, both the coarse graining and the fine graining very much have this generative model feel to them, right? You're trying to generate a complex structure. And the force field is very much like this property prediction I was mentioning earlier. Okay, let's talk about one more area. Let's talk about chemical reactions. One of the grand challenges in chemistry and in chemical engineering is how you can make everything. Well, maybe everything that's actually useful. There's an enormous number of things we could potentially make, and we actually know how to make very, very few of them. And particularly in pharmaceuticals, this is one of the big problems. There's a whole lot of things that people propose to make that we just don't know how to make. So there's a couple of interesting aspects of this in places where we're starting to see machine learning do something useful. So I'm going to be talking about, so what I mean by chemical reactions here is I'm starting with separated molecules. Somehow they're going to come together and form other molecules. Here I'm showing methane combining with oxygen to produce carbon dioxide, oxide, and water. So this is the kind of thing we'll be talking about. Now, as an illustration, I'm going to talk about this thing called Kumada coupling. And so basically what this is saying here is R is the standard representation for some set of stuff here in these chemical reactions. So here I have some R group in red over here attached to magnesium and then something like bromine. And then I have another one that's attached to the bromine. I put these guys together with some catalyst and various conditions. And what will happen is those two R groups will end up connected to each other. And this is forming a carbon-carbon bond. So reactions like this have been described in the literature. People spend a lot of time trying to discover reactions that can actually proceed, and it's a very, very interesting and challenging thing to do to discover these kinds of reactions. Let's talk about a simpler problem first, which is just even if I have this, how do I actually apply it? So here's an example. So here is something that matches that Kumada coupling template that I just showed you. So there I've kind of marked off what those R groups are. And so you would think that if I did what this thing says, that this matches the template, then I should get that molecule that I've indicated there. But it turns out this doesn't work. This reaction will not proceed because that R prime group has this aldehyde on it. Now, you can say, well, okay, fine, let's just write down those rules. So we've got the general rule and we'll start writing down the exceptions. This gets very, very difficult to do very, very quickly. And people are actually trying to write down a bunch of these rules and all the exceptions and get all of them right. But recently ML techniques have been applied to learn these directly from the data and be able to discover things that people didn't even know, restrictions about whether reactions would work. But there's actually more here. You may notice there's something I did not tell you about this reaction, which is what happened to all that magnesium and bromine, right? Does it form that? Does it combine with the solvent in some way and wander off in some other form? This problem of predicting side products is something that's not well understood. It's not even well measured typically. But it can be very important if you're trying to do things like chemical reactions without purification in the steps, so you still get with all this other stuff wandering around. But there's even more, right? Which is, I mentioned, in passing, you have to worry about things like what solvent you put them in. Sometimes you have catalyst. You want the reaction to proceed at some reasonable pace, but you're willing to elevate the temperature to make it go faster. How long do you have to do it? These are all things that are actually important to predict for any practical system that's really going to use this kind of reaction prediction. So that was the problem of, if you have a template, really making sure you understand how you're going to use it and if it will apply. Let's talk about the problem of reaction discovery, which is, this is something, finding a new chemical reaction. There are two big strategies. One of them is really using these, physics-heavy approaches, use force fields, do a lot of simulation. What you're seeing in this video from two groups at Stanford is a simulation of this Urey Miller experiment where they were trying to recreate what they thought early Earth looked like and to see if organic molecules would form by adding energy. And so what's going on here is they're running this simulation, and you'll see they kind of force all the molecules together. This is like kind of forcing them to interact quite a bit. And what you can see is they're forming more and more globs. They start to form more and more complex molecules. So this has been, this is an interesting strategy to kind of do this. But when I talked to one of my colleagues about this particular experiment, his comment was, they used every GPU we had for like a year. So these, this is again a place where you start thinking, what is it that we can, maybe there are ways that we can approach this with some of the approximations I was talking about before that can improve these kinds of calculations. Doing things like finding reaction barriers, predicting rates of reaction is really an interesting challenge. Now, on the other side, the other main strategy to doing this kind of reaction discovery is much more kind of, I will say, traditional data mining or knowledge-based. The idea is, go look at all the reactions we know about. This reactant, this solvent formed, combined to form this thing. Look at that as a giant network. And then use some of these standard graph ML techniques, somewhat similar to the things I was talking about before, and then predict reactions that you think will be there, even though they're not in your current data set. And Mark Waller's lab has done some of this. And what they've shown is if they look at the literature up to some point, they can then predict a whole bunch of reactions that were later published. And someone saying, hey, I discovered a new reaction. So they've shown that they can, there are reactions here that they can find. There's still a lot more validation to do in this place. That's one example of one of the reactions that they predicted before, sort of from data before this was actually published. So let me talk about one other problem here in chemical synthesis, which is retrosynthetic planning. So before, we were basically talking about these problems of what will happen if I put two things together. But that's not where we started this. We started this as, I want to make something, I've got a goal. And so the retrosynthetic planning is saying, I have a thing I want to make, please tell me how I'm going to make it. It should be with stuff I can actually get. It shouldn't be too expensive. I should get some decent amount of this stuff. It shouldn't be too hard, and it should work. And there's a bunch of other constraints too. And so here you might start with something like this, say, how do I go about doing this? There are two basic strategies that are now present in the world. One is very much in this kind of expert system. I talked about these rule-based systems where you say, okay, here's the reaction template. I'm just going to go and I'm going to write down all the restrictions. I'm going to be very careful about how I note everything, and then I can use your kind of traditional tree search method to go and do this. And there are several systems out there today that are using this kind of method. The other thing that's gone on is to do this in a much more machine learning way. So here, you use those machine learned reactions I was talking about before, and you machine learn some heuristics during the actual search process, during this search through all possible, the whole tree of possibilities as you're trying to find your way from that thing you want to make to all the stuff you can buy. And they sometimes have called the system AlphaChem in reference to AlphaGo. It uses a bunch of very similar techniques, actually. And the interesting result from their work was that they had the system design a synthetic route, and then they compared that to published routes. And then they gave it to a new chemist who had never seen either and said, can you tell which one the computer designed and which one the human designed? And the chemist couldn't tell the difference. Now, there's still a lot to be done to say whether they're actually good routes. Maybe there's some subtle flaw. There's a lot of work still to go here, but I think this is a very exciting direction for this kind of work, and I expect a lot more to happen here. The big problem in all of this is really data. So in chemical reactions, this is very much a big data problem. That's why it's in a big font. Unfortunately, we don't actually have that big data. And a lot of what we have especially has this really bad lack of negative bias. No chemist likes to publish, I put these things together and nothing happened. You just don't see that paper happen very much. Usually it's sort of one part of and then I did something great. And so the negative data is very poor, and a lot of it is not encoded very well. A lot of it is trapped in various contract research organizations or in big pharma. So let me give you a couple of final thoughts here. A couple of caveats to start off. As is obvious, this is a really high-level view. I'm really just trying to introduce you to a bunch of the problems here. I've not covered everything that's interesting or all the work in all of the categories. So please don't treat any of this as being complete, but I think it's a pretty good overview. And if you know something about the area, you probably noticed that I didn't discuss something called molecular docking. This is really, I will admit, is my own personal bias. I don't like it, I don't trust it, so I didn't want to talk about it. The other point I make is I talked a lot about what's going on for molecules and not for materials. A lot of times very similar methods can be used when you start talking about these extended systems, crystalline systems, or somewhat amorphous system with lots and lots more atoms, but you end up with a few special considerations that I have not talked about here, but you'll see lots of parallels if you really get into this space. I now want to do probably the most dangerous thing I'm going to do today, which is I'm going to go make a bunch of predictions. And I know this is being videotaped, so someone can chase me down in a couple years and tell me whether I was right or wrong, but I thought it would be an interesting way to end this. So here are my predictions. For these kind of simple molecular property prediction, like energy and solubility and so on, I think these models I mentioned, you doing interesting things with the 3D geometries and encoding them in the neural networks are going to do some interesting things. I think all the other things people are doing to try to improve representations and network architectures for those kinds of problems, I don't think they're going to be a very big deal. I just don't think it's going to change things significantly. That's number one. Number two, I think these generative models are going to be a big deal, especially in the pharmaceutical industry. The pharmaceutical industry is constantly doing a search for chemical matter, and I think these generative models can really support that process and systematize it in a way that is not done today. Number three, I think that in coarse graining, we're going to start to see much, much better coarse-grained models for more and more systems by using ML. Maybe we won't see it for proteins, which is this really, really well-studied system. But as we look at other kinds of systems like polymer systems, et cetera, I think ML is going to do something really interesting. And the last thing is on reaction prediction. I think we basically have a data problem. I think we have a lot of the ingredients of the ML techniques we need, but we just don't have the data we need to build great models. So with that, as promised, there's a bibliography here. If you go to that link, you will see links to all of the work that I've talked about here, and I'll be happy to take some questions. So questions about, I mentioned this data problem, especially in chemical reactions. How can we get better data or invent better data? I think there are two or three interesting things going on. One is the idea of using various transfer learning techniques. So we can go do something like make a computational benchmark where we can make up the problems ourselves and train with a lot of data. And then we have a small amount of data specific to a particular problem. You saw a similar flavor in some of the protein work where, hey, let's take a bunch of data about families and then see if we can use that model in an actual application we want. So I think that kind of technique is going to be useful. In reaction prediction in particular, the interesting thing is the claim has been made to me that the lack of negative data doesn't really matter very much because almost everything is negative. So if you just pick random things that are kind of nearby, it's almost certainly negative, and you can just use that. Whether that's going to really play out to be true, I don't know. But these kind of techniques to guess things that that are nearby but are known to be negative, I think is an interesting technique. The other thing that's, I'll say, broadly going on as more and more lab work can be automated in various ways, I think it'll help both in the consistency and the volume of data that we get for these problems. So I'm actually quite hopeful that over the next 5-10 years that things will get much better. Yeah.
Are people using these techniques to explore the unknown chemistries associated with the origin of life?
So are people using these techniques to associate unknown chemistry associated with the origin of life? Not that I know of, but that is not a firm example that is not being done. I mean, I do know that one thing for the Urey Miller experiment, but I actually don't know of any follow-on work even to that from that group off the top of my head. But I wouldn't say it's not happening. Yeah.
Finished off by remarking about a couple of things versus
So on this first one of, especially for reactions, the things we don't put on there really matter, like solvents and all this other stuff. And even when we talk about binding, one of the things I don't like about docking, for example, is we rarely take desolvation effects properly into account. And so this is where I think a lot of the physics-based models sort of have some weaknesses, because there are all these things that they're not even trying to take into account. And my hope is that some of these more data-heavy ML-based things are going to be able to do better because of this. But that's kind of that's a supposition that will be that's an advantage, and I don't have the concrete proof for it. On the pharma question of the issue isn't just that I need a molecule that's going to do something. I need it not to do a bunch of other things, which all result in various harms to the patient, for example. I agree completely, and this is actually one of the reasons I think that ML is going to be really useful here. Because even medicinal chemists and pharma will say, look, given a molecule, we know how to make it more potent. Right? We're already good at that. That's not our problem, right? We can look at it, we can look at crystal structures, we can do some experiments, we can quickly get there. The problem is they do have to think about a whole bunch of things they don't want it to do at the same time. And my claim is that human beings are not very good at keeping that many different optimization functions in their head at the same time. And this is where machine support for that process could really make a big difference. So I don't actually think just for potency optimization it's going to matter as much. So I completely agree with you on that one.
Okay, thank you again, Patrick.
Thank you all.